Chestnut blight is a damaging tree disease caused by the fungal pathogen Cryphonectria parasitica. It causes a lethal or serious decline of sweet chestnut, and symptoms on infected trees include discoloured, fissured cankers, orange, pinhead-sized fruiting bodies, and if you look under the bark you can find buff-coloured mycelial fans. It's native to Asia, although it's been found in Europe since the 1930s, and it was first detected in the UK in 2011. When outbreaks are at an early stage or they're small, it's possible to eradicate the disease by undertaking felling of the infected trees and removal of the stumps. When infection is more established, eradication becomes much more difficult. And it's for this reason that forest research scientists are looking at an alternative, a biocontrol using a naturally occurring mycovirus called CHV1. Cryphonectria hypovirus 1. CHV1 causes an effect called hypovirulence, which means that it weakens the fungus and reduces its ability to harm the trees and to spread. So this virus is used successfully in Europe, and treated cankers can heal, changing colour from orange to black as the fungus becomes less harmful. Forest research scientists working in our quarantine laboratory at Alice Holt have confirmed that CHV1 virus will be effective under laboratory conditions on UK isolates of the chestnut blight fungus. And we're building a bank of hypervirulent isolates that we can use in the future. We're working with DEFRA and Forestry Commission colleagues to ensure that we've got the correct legal framework in place to allow CHV1 treatments to be undertaken on mainland UK. But meanwhile, we're working on a really exciting and pioneering project in Jersey where the regulations are slightly different. So this project is funded by DEFRA and it's supported by the government of Jersey, who have given us permission to undertake a field trial of CHV1 treating infected trees in Jersey on the island. This work began in January 2025 when we visited Jersey and identified some suitable sites and some infected trees. From these trees we took some samples. Using these samples back at the lab, forest research scientists were able to transmit the CHV1 virus to the local fungal strains. And then in 2025, working with the scientists and the officials in Jersey, we used these hypervirulent isolates to treat cankers in the field on the island. The way we did this was to make small incisions around the edge of each canker. We've just undertaken in January 2026 our first assessment 6 months after the treatment, and the results are looking really promising. The untreated cankers have grown in size, whereas the ones we've treated have stopped expanding. If this project continues to be a success, it will be a major step towards future biocontrol treatments in the UK, and it demonstrates a really strong scientific collaboration with our counterparts in Jersey. Although these are early results, they're really encouraging, and the future landscape of sweet chestnut trees is looking positive.